Here's an experiment that asks people to draw a scientist that shows the overwhelming misconceptions people have about what science is. Throughout the summer, I spent a lot of time learning about the diversity within science. The scientists are not limited to stereotypical lab coats and mixing chemicals. But also this summer, I did just that for the first time, and it was one of my favorite parts. I learned so much that I will share with you now. To set the scene, we're sitting in the botany department, and we're given a petri dish with a beautiful green culture that we know is some part of a lichen, which is made of fungus and algae, but we want to know which part, and even which species. Our mentor set out with us on a quest to find that information using DNA analysis in the lab. The first leg of our journey is to extract the DNA. DNA extraction essentially consists of mixing a bunch of chemicals that'll separate the DNA from the rest of the cell. Throughout the lab work, we learned the valuable skills of properly pipetting and avoiding contamination. Okay, so now we've got these terrifyingly tiny tubes of DNA that we're going to want to get sequenced, translated into A's, G's, T's, and C's. But before that, we need to make thousands of copies of the DNA using a technique called polymerase chain reaction, or PCR. PCR utilizes heat cycles and a natural enzyme that replicates DNA in cells. Pipetting such small amounts was actually pretty difficult and quite repetitive. And now comes a test. Gels are used to determine whether the PCR worked. The DNA is run through a gel with an electric current to create bands. If the bands show up, the PCR worked. And if they don't show up, it didn't work. But now onto that sequencing I've talked so much about. An outside company actually did this for us. We sent them the DNA that worked, and they are going to tell us the order and length of the bases A, G, C, and T that code for DNA. However, science is not perfect. So they weren't able to tell us all of the bases of the DNA, so we had to go through with a computer program to delete the areas they weren't so sure about to ensure we had the most accurate sequences possible. Because now, we're going to blast them. Not with a gun or anything. BLAST is a database. Connecting DNA to traditional taxonomy is a big part of modern science. So, scientists have created such databases to connect sequences to species. We just pasted our sequence into the search bar and looked for the closest match. So, did we find it? Yes. Our mystery culture is Coximixus simplex, a species of green algae. Thanks so much for joining me on my quest through one version of DNA's journey through the modern lab.